Hello, my name is Joshua Mugabi. I welcome you to Behavior in Living Organisms. Questions 1 to 5. Question 1. In an experiment, the ability of the rats to learn the routes out of the maze was investigated. 3 groups of rats were used. Each rat in Group A was put in a maze, and when it found its way out, it was given 1 pellet of food. The time taken for each rat to get through the maze was measured, and the mean speed of each group of the rat calculated. Each rat in Group A was put through maze 20 times. The rats in other 2 groups were treated in exactly the same way, except that rats in Group B was rewarded with 16 pellets of food, and each rat in Group C with 50 pellets. Part A. Giving evidence from the graph, state the type of learning behavior shown by the rats in the experiment. It is operative conditioning or instrumental conditioning, trial and error learning, associative learning. Trial motor activities gave responses which were reinforced by a reward. Association of the outcome of the response in terms of reward increased future responses. Associative learning efficiency was increased by repetition. Part B. Explain how each of the following affected the behavior of the rats. Number of trials made. After 3 trials, the greater the amount of food given, the faster the maze was navigated. Now, if you look at this graph here, these ones of graph C, they were given 50 pellets each on completion of the maze, coming out of the maze. And these ones in Group B here, each one was given 16 pellets. And when you look at Group A here, Group A, each one after completing the maze was given 1 pellet. Rats are mammals with well-developed advanced brains, nervous system, can quickly learn maze complex, and larger rewards increased reinforced rats association with rewards with the movement through the maze, provided increased motivation. So those ones that were given larger amount of food, they responded very fast with the increase in the number of trials. And those ones that were given medium amount of pellets, their response was also minimal. And those ones with the fewer pellets, their response was low. Meaning that the movement through the maze increased with motivation. Larger quantity of food provided increased rats' response, and higher motivation provided made the rats to learn the maze faster. So you can see that the more motivation, the speed increased with every trial. Less motivation, the speed constantly remained the same. And a medium motivation, there was also a medium increase in the speed with increase in trials. Explain how each of the following affected the behaviors of the rat. Food quantity. After the first trial, the more food provided, the faster the maze was navigated. Rats are mammals with well-developed advanced brain, nervous system, which can quickly learn complex mazes, hence greater memory. The more food provided made the rats to master the maze faster, requiring less time to find their way out. Part C. Apart from the factors investigated, suggest two other factors that can increase the ability of the rats to learn by the form of learning under investigation. 1. Making the maze more complex. High level of brain development or well-developed nervous system. Part D. How does the behavior shown by the rats in the experiments differ from innate behavior? Instinctive or innate behavior is inborn, not acquired during the animal's lifetime. Learned behavior, it is acquired during the animal's lifetime. Instinctive behavior or innate behavior is genetically inherited. Learned behavior is not inherited, but the ability to learn is inherited. Instinctive innate behavior is intrinsic, that is, also present in animals raised in isolation. Learned behavior is extrinsic. That is, it is absent in animals raised in isolation from others. Instinctive innate behavior is inflexible. That is, not modified by the development or experience. Learned behavior is adaptable. That is, capable of modification to suit the changing conditions. Instinctive behavior, unintelligent and automatic. Learned behavior may be intelligent and intellectual. Instinctive behavior is stereotyped. That is, performed the same way. Learned behavior is permutable, can change over time. Instinctive behavior is permanent despite minor modifications. Learned behavior is temporary and short-lived behavior, although reinforcement can make it less or more permanent. Instinctive innate behavior is consummate, is fully developed and expressed at first performance. Learned behavior is progressive, subject to improvement or reinforcement through practice. Instinctive innate behavior, only one stimulus is required. Learned behavior involves more than one stimulus. Number 2. The figure below shows the response shown by a hen and dog toward a visible food source when each was first starved and then placed separately in a transparent wire netting left open at one end, with the food placed outside the netting on the opposite end, which is closed. State the form of behavior shown by each animal. It is insight learning or reasoning. Two, mention 3 characteristics of the type of behavior identified in A above. There is use of information previously learned from other behavioral activities, recall of the past experience, to solve problems never encountered before. It involves higher centers of the brain. Uses intellectual ability like reasoning, knowledge, cognition, thoughts. It is perfected by training and rehearsal. C. Explain the behavior response shown by each animal. We shall start with a hen. This makes seven attempts to reach the food, but eventually fails. A hen has a poorly developed nervous system, less advanced brain, low memory capacity, thus reduced ability to reason or to learn from past experience, can only find its food accidentally upon escaping from the netting to reach the food. So here you can see that the opening is here and the food is here. The hen is in the net. Instead of moving out of the net and go and find the food, you can see that our hen came to the net 1, 2nd, 3rd, 4th, 5th, 6th, 7th. 7 trials. It is hitting the net so that it can reach the food outside, which is impossible. And because it has a low memory capacity, it fails to get the food because it cannot use reason. Now, when you look at the dog makes very few or 3 attempts and then eventually finds the food. A dog is a mammal with a well-developed nervous system or the brain, large cerebellum with a high memory capacity, hence capable of reasoning. Offers a few attempts to size up of the situation, reduces or suppresses its natural drive towards the food source, escapes from the netting so as to find the food. So when you look at the dog and the food is provided outside, it comes and tries to pass through the net 1, twice, 3 times. Now it uses its brain and it cannot pass through the net. So it comes this side and passes through the opening, then it goes and finds the food. The figure shows habituation to touch by a group of tube worms, branchioma. In this investigation, groups of worms were tested by brushing their protruding branchial crown. 2 separate experiments were carried out. In experiment A, the worms were stimulated so gently that they only responded by withdrawing slowly their tubes. In experiment B, the stimuli were sufficiently strong to excite a rapid response from the worms. Part A. What is the effect of the frequency of stimulation on the response of the group of worms in experiment A and B? From 0.4 number of stimulation causes high number of worms responding in both experiment A and experiment B. That is around 0.4, somewhere here. 0.4. So you can see that the stimulation causes high number of worms to respond. The number of worms responding is very high in both A and B. High number of stimulations of 28 causes low number of worms responding to both in experiment A and B. Now at 28 here, you can see that the number of stimulations, like 28 of them, the response of the worms decreased. You can see they are around here, and then another one is around here. The response is very low in both of them after 28 stimulation. Increasing the number of stimulation from 0.4 to 6 decreases the number of worms responding in both A and B. So when you increase the number of stimulation from 0.4 to around 6 here, you will see that the number of those responding goes on decreasing at those different points. And even this one, you will also see the number of worms responding decreased. Increasing the number of stimulation from 8 to 20/22 causes the number of worms responding to rise and fall, fluctuate in both experiment A and experiment B. So from 8 to around 22, there was a rise in both of them, B and A. There was this rise, there was this rise, there was this rise, there was that rise. But they were also the fall. They were falling. If you look at them at those points, they went on falling. There was a rise and a fall in both of them. Increasing the number of stimulation from 22 to 28 has no effect on the number of worms responding, causes the number of worms responding to remain constant in both experiment A, experiment B. That is from 22 to 28. You can see that the number of response remained constant in both of them, A and B. Increasing the number of stimulation from 22 to 28 has no effect on the number of worms responding. Part B. Explain Roman number 1 the differences in the effects of the intensity of stimulation on the number of worms responding in both experiment A and B. There is a gentle or weak stimulation causes high number of worms responding. This is because weak or gentle stimulation does not easily generate a higher frequency impulse. In receptors and synapse, resulting in too few receptors in few worms to adapt or fatigue, allowing more worms to continue responding. Strong stimulation lowers the number of worms responding, because strong stimulation are capable of generating high-frequency impulses in the receptor and the synapse, which may result in too many receptors and synapses to easily adapt in many worms, preventing transmission of impulses, and few worms show response towards strong stimulation. Explain the responses of the group of worms in both experiment A and B beyond 22 number of stimulation. This one here and this one here. In both experiment A and B, the number of worms responding remained low and constant. That is between 21 and 28 number of stimulation. High number of stimulation, repeated stimulation, generates a high frequency impulses. This causes fewer sodium ions to diffuse inside the receptor cells. While a higher frequency stimulation prevents the secretion of neurotransmitter from the presynaptic knob, exhaustion of neurotransmitter at the synapse threshold may not be reached, and no action potential generated, and no further response by the worm towards repeated stimulation. Part B. What advantages does habituation provide to animals? Avoids wastage of energy on either harmful or beneficial stimuli. Enables quick escape from danger. Repeated stimulation is ignored, preventing fatigue. Enables organisms survive in an environment which is with disturbing stimuli like smelly places, noisy area, etc. It helps the animals identify neutral elements in their environment. Animals learn to disregard unimportant stimuli, saving it from fatigue energy and time wastage. Lowers the animal's stress and anxiety on seemingly threatening but rather harmless or unimportant stimuli. Minimizes sensory motor demands on the animal in responding to virtually neutral stimuli. Enables the animal prioritize their responses, that is, they tend to significantly stimuli, ignore less significant ones. Helps the animal recognize important clues or signals and hence adapt to ever-changing environment by offering an appropriate response. Number 4. In another experiment, 2 groups of rats were previously laid and placed to spend some time in the maze before the experiment. When the experiment began, each group of rats were separately introduced into a maze from the start and allowed to run about in the maze. Group 1 were given food whenever they succeeded to exit the maze every day. Group 2 were not given food as they exit the maze until the 7th day of the experiment. The number of blind routes was made by the groups of rats in the maze as they traverse was recorded as the error score. The result of the experiment are shown in the figure below. Part A. Account for the errors made by each groups while traversing the maze. Group 1. Initially at day 1, around here, day 1, the number of errors scored by rats in group 1 was high. But scored in group 1 was really very high here. Errors scored. Because the rats had not stored enough information about the routes in the maze. Between 1 and 4 days, errors scored by the rats in group 1 decreased more rapidly. So from 1 to 4 in group 1, the errors decreased rapidly. And between 5 days and 7 days, error scored by the rats in group 1 declined rapidly, still from 5 to 7, declined rapidly, because the rats had stored rapidly much information in the brain about the routes in the maze. Besides, They were motivated by constant reward provided, making them learn faster and hence traversing the routes with reduced errors until no errors made by the seventh day. Seventh day, if you look at the seventh day here, they were no longer making any error on the seventh day. More gradual increase in error score by rats between 4 and 5 days arise from occasionally very few blind routes made by the rats during the experiment, from 4 to 5. That rise in the error score was due to occasionally very few blind spots made by the rats during the experiment. Group 2. From 1 to day 2, errors scored by the rats in group 2 remained constant. That is from day 1 to day 2. Here, they remained constant. Those are the errors. Then between day 1 and day 5, the error score by the rats in group 2 declined gradually. That is from day 1 or day 2 up to day 5. There was a gradual decline in the errors. The rats are slowly storing information in the brain about the routes because they were not motivated by any reward, and they consequently learning slowly to successfully traverse the route. From 7th to 9th day, error score by the rats in group 2 declined. That is from 7th to the 9th day. 7th to the 9th day, the error scored by the rats in group 2 declined rapidly because the rats are using much information stored in their brain about the routes in the maze to traverse it during the exploratory learning. Between 5 and 6 days, error scored raised gradually by the rats in group 2 due to few occasional blind routes made by the rats during the experiment. So from here to here, there was increase in the error score due to occasional blind routes made by rats during the experiment. Part C. From the information in the figure, state any 3 ways intelligence of an animal can be determined. The duration or time they taken to stop error scores, with or without rewards. 2. The rate at which the error scored declined, with or without rewards. 3. Duration it takes to remember and accomplish the task without trials. And 4th, complexity of the routes in the maze is transverse. Number 5. During experimental trial, a cat was placed inside the puzzle box. If a cat pulled the loop with its mouth or a paw, the door opened and it could escape. The time taken for the cat to escape was recorded. The experiment was then repeated several times with the same cat. The figure shows the graph of the time taken for the cat to escape from the puzzle box during repeated trials. Part A. Account for the changes in the time spent by the cat in the box at different trial numbers. Time spent by the cat is highest at the first trial. Here, it is highest at the first trial. The cat spent a lot of time at first in cage since it had not learned how to escape. Environment was new and still so complex. Cat investigated the puzzle box by exploration, pulled the loop accidentally, by chance, or by trial and error. A reward reinforcement, for example, food, led to escape. Time spent in the box decreased as the number of trials increased. Due to learning, rapid decrease in time spent by the cat in the first six few trials here. Greatest change in response occurs. The cat is highly motivated, caused by the food, which is the reward. Yes, thus positive reinforcement, improving the performance through error rate, though error rate still high. From trial 6 to 20, around here, 20, there was a gradual fall in response time. Time spent in the puzzle is very low. So from that time, you can see that the time spent in the puzzle is really very low. This is because learned rapidly to associate escape with the reward. It pulled the loop sooner due to learning, easy to remember, sufficient acclimatization period, repetition enabled learning to occur. Time spent by the cat in the box fluctuates. Rise in time is due to forgetting. Fall in time is due to learning. Needs to make choice, as learning is by choice. Where we are seeing some rises in time due to forgetting, comes back, is remembering, forgetting, like that. Part B. What evidence shown in the figure shows that learning took place? Errors reduced with time. Time spent in the box reduced with increased number of trials, and time spent in the box fluctuated. State 3 factors that could affect learning of the new situation, like a maze in animals. 1. The size of the reward or punishment. 2. The state of development of the brain. 3. The state of development of the sense organs. 4. Complexity of the maze or the puzzle box. Part D. How could the time needed by the cat to escape out of the puzzle box be reduced? By rewarding it at the end. The right attempt reduces the time required for it to leave the cage, giving it food. When it makes a wrong choice, the cat is punished, increasing the number of trials per day to enable learning, reducing the complexity or number of turns in the puzzle box. Part E. Define operative conditioning. It's a form of associative learning through trial and error learning, where the animal learns to associate one of its own behavior with a reward or punishment, and then tends to repeat or avoid that behavior. Part A. Discuss the characteristics of reflexes. They are heritable, encoded in DNA, passed from generation to generation. They are intrinsic, present in animals, raised in isolation from others. Stereotyped, performed in the same way each time by each individual of a particular species. Inflexible, it's not modified by development or experience. Consummate, fully developed or expressed at the first performance. Unintelligent and automatic, does not require learning or conscious thought. Conditioned, can be modified, produced following exposure to a new stimulus. Thank you very much for participating in behavior in living organisms. Question 1 to 5. Please do like, share, and subscribe.